Hi, this is one more video series. Now we are going to cover biotechnology. In the previous thing we have done, we have seen basics of the space technology. Here also we will do some basics of the biotechnology. I'll try that once these basics are clear, when you go through any current affairs and basic books, you will be able to understand things. See, in science and technology, there will be certain core concepts. When these core concepts are clear, rest of the things are very general, very easy to understand. There is nothing more conceptual, theoretical beyond this. As I always say, we need to understand science and technology as a generalist, as a layman, okay, as a some good knowledgeable person, not as a kind of expert. We don't expect here to be experts here, okay. UBS is not expecting us to become do some kind of R&D activities, okay. So that just as a good knowledgeable person, you have to know basic understanding of what is happening. In the same way, we will try to understand the biotechnology also. Now, before going into the biotechnology, see, with the recently corona in the news, you also need to see from the basics of the biology, types of the blood, okay, antigens, antibodies, types of the immunities, and vaccines and all. Okay, just I want to take one topic of immunity system before getting into the basics of the biotechnology. See, uh, we have now currently the major issue is lack of immunity to the coronavirus, right? So immunity becomes very important topic. No immunity can be from not active, it is acquired. So there are two types of the immunities: acquired and this acquired, that is active, and this is passive. Now this one innate immunity, you have got it by birth. So whenever a kind of uh, some kind of infection happens, your body has already immunity developed to it, so it will immediately act immediate and destroys the bacteria. So this is the innate immunity by birth. Let's say there are certain new infections that has come into the body. Now this does not work when your body slowly recognizes that immunity, recognizes that infection. Over a period of time it develops the immunity. Let's say now we are talking about the herd immunity. There is nothing but they are talking about this thing only, okay. Now your body is able to generate immunity for that, and it can fight a bacteria, but it will takes time, slow process. So the first time you are infected, then body will identify this kind of infection and develop the antibodies to fight it. Next time when when the infection comes, these antibodies are already there, it will easily identify the invading agent, okay, and it will attack. So this is a kind of acquired immunity. It is not there by birth, but it has slowly developed. Now in this slowly development, it can be active, okay. Normally, as I have mentioned till now, your body has developed that kind of immunity in a slow process. Now there is a kind of passive immunity. In this passive immunity, you are given certain outside support to develop such kind of immunity. One thing here is vaccine you can be given. Now vaccines, there are different types of the vaccines which you have to again go through these things separately. So that I'm just giving you here a broader framework from where different things can be fit in, so that you can get a broader idea. Now there is one more thing called uh, immunotherapy, okay. Immunotherapy, here you might have heard about see monoclonal antibodies, okay. This you think you might have heard about in this news. The people are they are developing these things, and you might have also heard about the plasma therapy and all. You see what is happening here is, here the antibodies are cultured in the lab and they are given to your body so that your body is able to fight this. So this is you are getting from the external support. Now currently they are working on this also. Now apart from this, there is active immunity. How there are kinds of active immunity? See, now in this there are different cells, different cells are involved in this immunity. Now, what are the different kinds of cells that are involved in the immunity? Let's say in the innate immunity, the cells involved here are, let's say, neutrophils, phagocytes. Okay, there are the dendritic cells. So various cells are involved here in this immunity. And in the same way, here in the acquired immunity, you might have heard about different cells called white blood cells. You might have heard about. Okay, in the white blood cell, you might have heard about the lymphocytes. Okay, lymphocytes. In this, you might have heard about these things. You have heard about the T cell. Okay, B cell. Okay, now they can put different kinds of questions here. Okay, the lymphocytes are nothing but they are part of the WBC. Within this, you have T cell type of immunity and B cell type of immunity. This thing, B cell type of immunity, this thing, they generate the antibodies to fight the infection. They generate the antibody. Okay, this thing T cell, they don't generate the antibody, they use cell mechanisms to attack. Okay, as a defense. So these are things. Now wherever the lymphocytes are present, they are called lymphoid organs. Now they are present in different parts of the body. Okay, let's say they are present in the spleen. Okay, they are present in the bone marrow. Okay, so different places they are present. Now this is a broader understanding. Now see, for the cancer, currently, let's say it was in the news. Okay, they are using chemotherapy where they are trying to destroy the cancerous cells. Now because of this also, this immunotherapy is in use. What is happening in immunotherapy is your own immune system will be strengthened to fight the cancer cells. During the times of the cancer, what is happening is your immune system has been muted. A kind of protein will be generated by the cancer cells. This will mute your immune system. So now through various mechanisms, they are trying to unblock this kind of blockade that has happened to your immune system. So they are trying to strengthen your immune therapy system. Okay, in this they are also strengthening monoclonal antibodies. They are cultured antibodies that are taken back into the body, or else whatever the blockade that are faced by the T cells and B cells, they are trying to unblock it. Why? Because T cells and B cells will attack either through antibodies or just through the cell systems. Now, but what cancerous cell will do? Cancerous cell will block them. Okay, it will these two cells will not recognize, it will not recognize any kind of problem that has been entered into the body. Now through various mechanisms you will regrow it. Okay, so I think these whole process, whole update is clear, right? Now even in the plasma therapy also, what is happening is, see, just like active immunity, your the plasma is nothing but the that is present in our blood, I think, right? Okay, so they are developing a kind of immune system. Now these antibodies, they are able to fight the virus. Now these antibodies will be cultured into your body. So whatever the monoclonal or else what is the plasma therapy, still studies are going on, and these are very slow processes since these are based on the active immunity. Okay, since these are slow process, they want to go to the passive immunity. Okay, the vaccine phase where the things are generally acting very faster. Now while developing these things, now this vaccine and all is in the news. That's why this was also in news. Controlled human infection model, where you are deliberately given some kind of infection and then vaccine is tested, so that this process will give you the quick results. So with the corona also, this is in the news. Even earlier they are also trying, or generally what happens in the clinical trial models. Okay, the vaccines are given to the large mass of the population. Okay, they generally wait till some infection happen, and from that they can get the studies. This is a slow process. Okay, this is a fast process. So even the developed countries, most of the developed countries use this model, and currently India is not using this model. So Department of Biotechnology is asking, so think, think, bhaiya, think, socho. Why not they try this method? Okay, slow, fast result, bigger and all. So this is the one broader aspects which you must know. So easy, right? Things are easy, nothing difficult. Now, even we'll go to the DNA, RNA things. Okay, things are very easy. Just pay attention here. Okay, see now to understand the biotechnology, first you understand what is this biotechnology. Biotechnology is nothing but the utilization of the living organisms for the some kind of benefit, well-being, anything for some kind of research purposes. Let's say we make bread, we make curd. It is using of the yeast, right? Bacteria is being used, okay, and then cultured. Now we are having the curd and all is happening. Okay, this is also biotechnology. This is called the traditional biotechnology. Traditional biotechnology. Now we have one more thing called modern biotechnology. In the modern biotechnology, you will alter, okay, uh, you will change the genetic structure. Genetic structure has been changed. Okay, this is called the modern biotechnology. Let's say BT cotton, okay, and then BT brinjal, mm, mustard. Okay, so all these things, the genetic structure is being altered so that we can get a better variety. Let's say one thing. Okay, there is a kind of let's say this is different from the hybrid thing. Okay, well, let's say now, let's say one of your friend wants his he's so much fascinated by, 1 second. So he want his baby to be something look like some foreigner. Okay, something very fair. Okay, some kind of different kinds of eyes and kind of silky hair and all. And at the same time, he also wants uh, Indian kind of some, let's say, immunity, or else yeah, that's what generally it is believed that Indians have bit more immunity than the here temperate countries, nations. Okay, so he thought that okay, I am from South India. Okay, let's marry a girl. Okay, who is fair and have soft hair, silky hair. Okay, let's marry her. Okay, so this is what he has done. Okay, now this is kind of a hybrid matlab, कुछ ज़्यादा ही होगा मतलब in terms of the humans, but still I'm trying to explain it. So now they married and they got a kids, yeah, but to his surprise, he got um, South Indian kind of futures. Nothing bad per se, but this guy is wanting foreign kind of things. Okay, he has got brown color, maybe black color, or we generally don't have that kind of silky hair, maybe moderate curly and all. Okay, whatever the South Indian features we have, brown and all. Okay, he has got that, and immunity wise, he has got this ones. Okay, whoever they are generally they have the low immunity. So all the bad things which he, according to him, things he has acquired it. So this is what yeah, when you go for some kind of hybrid things, even with the crops, this is what you face. Okay, then this person thought, okay, he told to his friend, Dek beta, I have faced this thing, you go for another method. Then this person thought that okay, when I have my baby during this let's say excel, okay, embryo. Okay, here this is the embryo. Okay, one cell. Okay, there is one cell. Later on, this person it will become a human being, right? So during time of the embryo itself, in the embryo, there is one cell. Based on the information that is stored in your cell, that is genetic information, you will be developed into some kind of human. Okay, now I'll alter this kind of genetic information. Okay, I will give, let's say, Ritik Roshan body. I will say I give Aishwarya Rai kind of eyes. Okay, what more Amitabh Bachchan kind of height. Okay, Sachin Tendulkar kind of batting things and all. Okay, Vijay Devarkonda, WhatsApp guys, kind of some guys कुछ और someone else says. Okay, so these kind of features I give, and modifying by modifying his genetic structure. Now this is where genetic technology comes in. Okay, he's modifying the genes. So you might have heard about the designer babies. Okay, this is the concept of the biotechnology. You are so that the results are fast, okay, accurate. You are wearing the changing the genetic structure. Now this genetic structure can also be will also be transferred to his next generation. Okay, his kid. Okay, so that is why there is a reason risky. It can also be mutated in the process, and this person can die. Okay, and you don't know what like say there are so much of the dark DNA. Okay, dark DNA means we could not study this kind of DNA, but there are certain impacts of this dark DNA. Okay, so we don't know all these things, and the person can be affected. We don't know what is happening. So the same thing when during times of the HIV, where a person has used some genetic treatment, the Chinese person to give us some kind of treatment by affecting the genes. It was in the news. Thing is clear. Now we will get into the basic things. So first thing is, what is DNA? Then we need to know what is RNA. Okay, so to understand these things, we need to understand where they are present, why they are present. Okay, what are their functions and all. Now within the cell there is a nucleus. Now within this nucleus you have the chromosomes. Okay, chromosomes, they have some kind of structure like this. Okay, on these chromosomes you have DNA. Okay, cell. Within the cell you have the nucleus. Within this nucleus you have the chromosomes, and then with on these chromosomes you have the DNA. Now what is this DNA? So these DNAs are based on the nitrogenous base pairs. Second one, phosphorus. Third one, sugar groups. So all these things become a kind of monomer, nucleotide. Okay, when all these monomers, let's say this was m1 plus m2, all these things, when we add whatever the things we have, then that is nothing but the DNA. Now this DNA, when we same thing, when we properly put in a structure, this will be a kind of double helical structure. Okay, these are nothing but the phosphorus and sugar groups. We don't have to get into them. Okay, all these are attached to the oxygen and hydrogen atoms. Okay, carbon atoms are there. So that thing you must understand. Okay, so so these nitrogenous base pairs are in the form of ATCG. Only these four will be there. These are the DNAs. Okay, so DNAs are nothing but the first one. They are the monomers. Monomers. New. Okay, all these monomers together form the DNA. Now, what is this single monomer having? This thing. Okay, these three things. A single monomer will have nitrogenous base pair called ATCG, and they are attached together by means of phosphorus and the sugar compounds. Okay, so you will have carbon, hydrogen, oxygen, these things under this. So within this, let's say ATCG, it is nothing but a kind of code. Okay, then again you have ATCGG again. Okay, CG. Okay, TA, something like that. Okay, all these things have a code. Okay, three polymers. Okay, these things together is having a kind of code to generate a kind of function. Then this thing is called gene. Within this, let's say this part has having specific function. Okay, let's say this part talking about this person should have silky hair. Okay, this part is saying this person okay, should be liking these kind of foods. Okay, this part is talking about he should be having allergic to this thing. This part does say that he should get in these situations headache. Okay, this thing, like this. Okay, so all these things, the single part, it can be, let's say we have maybe billions of such kinds of nucleotides. Okay, so within this one thing, one or two, anything, few things constitute a gene. Okay, which is deciding, it is nothing but a kind of code. Okay, this code will give that kind of a function. So based on, based on different of the codes that are present in the body, you will be having, this is nothing but the software. This is a complete software. Complete software is DNA. Okay, individual software. Let's say this is for gaming. Okay, this is for your Microsoft. Okay, this is for programming, something like that. Okay, let's say in the same way, eyes, ears, nose. Okay, how the shape of your nose, how your body color, how your hair color, hair structure. So all these are the genetic information. So coming back again, you have the cell. Within the cell, you have the nucleus. Within the nucleus, you have the chromosomes. On the chromosomes, you have the DNA. Within this single, within this one part, few specific things will give are constitute genes. Now, what they can ask you, what are the components of the DNA? Then you can talk about the deoxy ribose sugar is present. This sugar is a deoxyribose sugar. Okay, and then you have the phosphorus component, nitrogenous base pairs. These are all these things constitute a one kind of monomer. Monomer means single. Okay, when the all these things there are, let's say, 100 formed together. Okay, then or else more than the two or three, then they becomes a polymer. Okay, all of them together is present in the DNA, but each of the, let's say, few of the polymers combine to form you one specific gene which has some kind of functionality. Clear? So now, how you will behave, what kind of diseases you will get, okay, what kind of allergies you get, what kind of features you have, everything is based on the genetic information stored in the DNA. How do you get this genetic information? You will get from your mother through the egg cell, and also from the father through sperm cell. Okay, uh, so so through this genetic information, you will be whatever the data you have coding, you are getting. So in your DNA, you have different number of genes, right? So each gene is having different kinds of expression. Now this is present in the nucleus. Okay, within the cell there is a nucleus. Okay, now here, this is a, let's say this is the nucleus. Now outside the nucleus there is a ribosome. Now this is a factory where this coding will be expressed. This is nothing but the gene expression. So here you have the genes. Now these genes are transferred to this coding area, ribosome. Here actual proteins will be taken place. Okay, now this person should be having black hair. Where that black hair is manufacturing, it is being manufactured here. So when this you will get the black hair, then this is called the gene expression. Let's say you have so many things, but not all the things are expressed. Okay, gene expression does not happen. Only when this has been transferred, then there is a gene expression. Now who is this person that is transferring? That is done by the RNA. So these things are in the totally in the news, right? DNA and RNA. So what is this RNA? Okay, so most of your genetic information is stored in the DNA, not in the RNA. But there are certain virus, let's say coronavirus, who has information stored in the RNA. This is one thing, and it is a single stranded structure. Okay, and it has same nitrogenous base pair. Instead of ATCG, you have the U, right? This one is not there. Okay, and it has and instead of double stranded structure, it has single stranded structure, and the complexity is less. Okay, complexity is less, and it is helpful in the transfer of genetic information. Okay, transfer of genetic information from here, from the nucleus to the outside the nucleus at the ribosome. That is called a mRNA, messenger. Message gives the information. Okay, tRNA. tRNA. There are two types of the RNA: messenger RNA and tRNA. And this tRNA is actually bringing the information. This mRNA is saying these are certain, these kind of genes are there. Okay, and tRNA the protein, let's say for the protein there is a need for the amino acids development, right? This tRNA will be bringing different amino acids necessary for formation of this protein. Okay, and there is one more rRNA, that is this is present in the ribosomes for the doing this kind of activity. So now coming back to the again one thing, within the nucleus you have the genetic information. Outside the nucleus you have the ribosome where the protein manufacturing takes place. From here the information goes to here through the help of the RNA. Okay, now not every genetic information is being transferred to here. So the what kind of gene expression you have, it will depend on the RNA. So there are also various studies that are going on in studying the various things that are present in the RNA. Okay, now you might have heard about the what's called transcriptome. Okay, now there are so many mRNAs, right? Okay, which are transferring information. Just like all the six genes is becomes the summation of all the genes becomes the DNA. Here all the summation of the RNA becomes transcriptome. Now there is one more process called the transcription. Okay, these were already asked, so that's why I'm highlighting this. Transcription, translation. Okay, transcription means the process through which genetic information has been transferred from the nucleus to the ribosome. Translation is the process through which genetic information is transferred into the protein. So we have talked about the transcriptome. Okay, transcriptome. Okay, this is nothing but the sum total of all the RNAs, messenger RNAs. Okay, so this is called the transcriptome. Okay, so you have got the information. Now this is how. Now let's say the why. So let's say there is a human genome project. Human genome project. We are studying the whole DNA sequence of the human being. No, why do you study the whole genome sequence? Okay, let's say this gene is not properly working. Okay, or else let's say you will understand. We have now come to know that 95% of the genetic information is almost same in the plants, animals, micro, any kind of organisms. Okay, so that is the reason we can have the transfer of the genetic structure from the animals to the plants, plants to animals and all. Okay, or else it is not possible. And we have a very close resemblance of the genetic information with the monkeys and all. So we can know the evolution of the species. Okay, we can also know, okay, functionality of different species, different genes. Now when we know the functionality of the different genes, let's say this gene is not working properly. Okay, we can silence it. Okay, or else we can use it in a certain way. Or else let's say this gene has very good property. Let's say one of you is having a property called where when you study once you will not forget it. Okay, let's take out the gene and give it to the all the students. Okay, that's how we will study the different gene. We can also study the individual genomes. Okay, let's say indigen project that is happening by the CSIR. Okay, CSIR, where few Indians genes we are studying. Okay, to study how what their genetic structure and all, to understand these things. Indigen project. Now this is the current affairs, right? Now you understand these things. Okay, you can easily correlate with this thing. Now you can easily correlate human genome project. Okay, they are nothing but studying of the genetics, some sequence of all the genes. The same way there is a project on the RNA sequencing also. Okay, so now these things will become easy. Okay, Manav Atlas. Okay, where they are not going for new studies, new research. Whatever the existing information is there, they are trying to find out through this information the different network connections between the different tissues. Okay, okay, what is the kind of networking that is there present in the tissues? This is a some basic information I have tried to convey to you. So now this thing is clear for you. Now you know DNA, now you know RNA, you know the ribosomes and all. Okay, see, now this is a nucleus, and here all the genetic information is stored. Here the ribosome, here the RNA. factor is happening. Now remember one thing here, whatever the information, how you grow up, how you have all these things is present in your nucleus. For every one of our body we have number of cells, right? In every body part we have the this kind of cells and nucleus structure everywhere. How we grow, how we develop and all, it is there in this stored in the genetic information. But apart from this outside also there is a DNA present. Okay, this DNA is not about the entire structure, how it will have. Okay, let's say there is a mitochondria. Okay, how this mitochondria should function and all. Okay, how this mitochondria should generate the energy and all, it is developed. Okay, so these are certain basic things you must know. And also see what are the other organelles that are present outside the nucleus. And there are and let's say there are two types of the cell structures. They are called eukaryote and prokaryote. Prokaryote, okay, spellings you please check it. This one prototype. Okay, so these are less evolved. Okay, these are most evolved. Okay, first time amoeba, unicellular animals. Okay, more less evolved animals are prokaryotes, and more evolved we are the eukaryotes. And there is also cell wall present for the plants. The cell wall is present. Okay, yeah, between outside the nucleus, separating this membrane structure and all. So just see certain basic things, that's all enough. Not more than this. Organelles, some basic things: Golgi complex, lysosomes, two or three more things that you need to study. Not important, but these are the very important thing you need to know for the biotechnology, and recently for the coronavirus also, that you need to know. Now let's move. Okay, certain other things, how these things are now applied. Now see, so now we have understand the importance of the genetic information, okay, present in the DNA. So now the modern biotechnology is talking about modifying of this genetic structure and making use as per that. Okay, now let's say this is a kind of genetic information till here, one kind of information till here, one kind of information till kind of one information. So now what can we do? Okay, let's say this is a faulty gene. Okay, what we can do is, now see what is happening from there is a genetic information from here. It is from here it is being transferred by the RNA. Okay, to RNA, and then genetic expression will happen. Okay, now let's break this connection. Okay, this is called gene silencing, but still the gene is there. Later on you can again revamp it. But what happened, let's say you're removing this part itself. No, this is called a genetically modified organism. Or else you are enhancing this gene through adding one more feature. How will you do that? Okay, this is called recombinant DNA technology. You might have heard this. Okay, now recombinant DNA technology. Okay, I cannot just take the scissor and cut it, right? Okay, you need some kind of enzymes. Okay, through enzymes you will through you will cut it or else add it. Okay, so there are certain steps involved in it. First thing is, see, these are the four things. You will use the enzyme to cut a part of GNA. You will cut it, and then let's say now you have a we are going for the BT cotton. Okay, this BT cotton, nothing but bacterium thuringiensis. Okay, in one of the bacteria you have a kind of protein. Where this protein is nothing but releasing a kind of toxin which can attack the pest. Now this toxin, if you transfer to the crop, then it can be a better thing. How you will do? You will use the enzyme to first to take out the gene. You will put in a kind of vector. It can be anything. You can transfer it to the virus, and then you will transfer to the crop. Now ligase is nothing but the attaching thing. Okay, it's kind of gum. Okay, you are attaching it in and putting into the host organism, then these things will multiply. Okay, this virus, the whatever the wherever the cell is needed, it will go and it will attack, and it will become part of the genetic structure. Now see, you have been modified the gene which earlier was a part of the bacteria. Now it is being transferred into the cotton. Why this is able to transfer, do such kind of things? I told you 95% of the genetic structure is easy, same. Okay, you can transfer from bacteria to the plants, bacteria to the animals, animal to the bacteria, anything you can do. Now they are also trying to do beta carotene, golden rice. Okay, beta carotene part in the rice they are trying to increase. They are also trying to make your let's say now through PDS system we have only rice. This rice in we say that it is not a nutrition rich. We can push this rice with the vitamin A and then also make it protein dense. Okay, that thing you can do. And also currently we have the droughts, we have the floods. Now make this crop flood and drought resistance. See, these are the things that were asked in the there was a question importance of the biotechnology in agriculture. No, these are all the things. Okay, now in the same way there is a things of they are thinking for the BT brinjal. Okay, BT must not BT. Okay, this is something mustard. Okay, for the mustard also they are trying. So but cotton we don't eat, right? These things we will eat. So there is a need for very important evaluation before doing any kind of genetic modification. How we do the genetic modification? This is the process. Okay. And there is recently one more process called the CRISPR Cas9. This is nothing but simply a kind of process. Cas9 is a kind of enzyme which will do the cutting, and process is almost same. So BT brinjal, this is genetically modified the brinjal, same bacterium thuringiensis. This will reduce the need for the pesticides. But what is happening over a period of time? Bacteria is achieving that resistance to the pest, and you need to go for the next higher version. Okay, so for cost of the input seed is increasing for the farmer, and when you consume this pesticide kind of a modified genetically modified brinjal, we don't know what will happen in our body. Different bodies have said different things. Let's say there is a farmers organization called Shetakar Sangaram like this. Okay, they said that we need to go for this genetically modified organism, which is currently having the ban. Which is the body that is taking care of this approach, genetic engineering appraisal committee. You need to see under which body it is there. It is under the Ministry of Environment Affairs. Okay, that is a statutory body. You need to just see this thing. Okay, a little bit. It was also already previously asked. Now, what is happening in the mustard seed is this is first time genetic modification done by indigenous technology completely by the Indians. Okay, Delhi University. What they do is the mustard seeds are generally self-pollinating. Okay, what they did is they use some bacteria to make this plant infertile, and they have been they want to I know only this diagram, so that's why even for the plant and human I'm using the same diagram. Please bear. Okay, they want to hybrid this with a European variety. When they have used again genetically modifier of this organism, so that now this one is unfertile, but when it mates with it, this unfertile things again go, and again they can do mating and they can develop the hybrid variety. This is a genetically modified thing developed by the Delhi University. So same thing. So what are the problems? So environmental problems. Now it can reduce the biodiversity. Now since after the BT cotton, a lot of local and traditional varieties has been lost. When you consume that, we don't know what kind of impacts that will have. Okay, there are pros and cons. They are not much relevant for your repellents, but you need to have some broader understanding. It is easy for you to understand once you know these things. So you know why it has been used. Okay, for the production, to check the pest, for the increasing nutrition value, for the floods and droughts. Okay, and this is the process. So now these are the certain things. Even they are used in the industries. There are number of applications out there in the industries, in the agriculture, in the health and all. But concept is same thing. Now you can easily understand when you read at the different concepts. Now we will move into the human aspects also, health aspects. So there is a one more thing you need to know here is a DNA profiling. Okay, DNA profiling. DNA profiling is done for the individual. Okay, individual. This is different from the human genome project. Okay, for the human genome project, generally whatever I told you, 99%, 99.9999% of your gene and my gene is same. Okay, only one thing is different. That is that individual have variation in their genes. So that thing can be found out by the DNA profiling. Let's say I have murdered any of you and have escaped from there, but by mistake I have left my uh, hair there. Okay, मेरे बाल ज़्यादा झड़ते हैं, and then I missed my hair there. Okay, and then you have got my hair, and through other information and all, they have also stored my even DNA database, which recently they are talking about the DNA profiling bill. Now now this hair strand through this they will get the DNA structure. Now they will use the supercomputers to match it. Then they will find Aadhaar. Okay, this guy has been doing this. But what will happen? So this is some advantages we have. Okay, but what will happen? Just like a movie twist, and that is not my hair because of some hair. The hair has been fallen there from beside street, but the actual person's hair has been not there. But these people have caught me and arrested. So because of this reason, DNA profiling is being not being passed. Okay, and so these are not the foolproof mechanisms. So I think this concept is also clear for you. And there is one more thing that is also in the news, that is called these things you need to know: cloning. Okay, cloning means identical copies. Okay, let's say you are there. Okay, same ditto like you, hello brother. Okay, everything is exactly you. How it will happen now? How do I form? Okay, how do we form generally? Okay, let's say there is a embryo. Okay, this it is kind of cell. Okay, when two cells come together, egg cell and sperm cell come together, then you will form the embryo, right? um, Now within this embryo, so development will happen in this. The genetic information is taken from the mother and also from the father. Now let's say from this, within this there is a nucleus, right? Within the Excel there is a nucleus, and here this kind of information is being stored. Okay, both of mother and father's information is stored. Okay, let's say this is some other kid. Okay, so kid is being born. Okay, because of the mother Excel and father sperm. Okay, new baby is going to form or something like that. But I have removed this nucleus structure. Nucleus is where you have the genetic information, right? From that nucleus structure I have removed, and I have transferred my nucleus here. Okay, now this one later on fertilized, it will develop into a human. That is nothing but me, because here in the nucleus, before fertilization I removed the nucleus. A cell has been developed. Within the cell, the nucleus which has having the genetic information, this thing has been removed, and I have placed my nucleus here. So the next person will be hello brother. He will be same like me. Okay, he will be the exactly growing like me. Okay, now cloning, as you know, as usual, they can have ethical issues. Okay, now let's say suddenly tomorrow you will see again Narendra Modi. Okay, but it will take to grow and all. But and in between transferring also there are certain problems. This cloning is exactly not happening and all because of the ethical issues and all, various constraints and all. They are not going ahead with this thing because act of the nature and all. These things all useful for your GS4 also. You need to have some kind of opinion there. This is one thing. Let's say somatic three parent baby you might have heard about. Okay, three parent baby. Yes, I'm explaining all the concepts. It's simple, easy concepts, right? Now let's say this is the Excel. The mother and father has come together, and then within the Excel you have the nucleus. Okay, this is mother's Excel. In this nucleus you have acquired the genetic information of the mother and father. Now this Excel will grow, right? This embryo is nothing but the Excel. Okay, these things will develop, and later on the baby can have form. But this mother, I have told you, the DNA is also some data information is also present there. Okay, let's say she has the kind of mitochondrial defect she is having. Okay, when this person grows, okay, baby grows. Okay, this person is acquiring the, since this is the Excel. Okay, this Excel, within this Excel there is a mitochondria. This is having defected one. Okay, so now this person will also have defect in his mitochondria. Okay, now what we can do, we take another healthy mom, Excel. Okay, now you transfer this nucleus into the healthy mom. Okay, now she has a healthy mitochondria. Now fertilize this thing, and you will have a developed a baby which is having healthy mitochondria. But mitochondria come from the third mom. Okay, third parent. Okay, and nucleus has come from the mother and father. So this is a two parent baby. Again, ethical issues and all, the same thing. You can, now you can look into the those conceptual things and you can understand. Nothing easy, very easy, nothing technical, nothing something rocket science. If you have done rocket science, even I would not understand. Very very simple terms. If you understand these things, simple things, you can easily eliminate the options and you can arrive at the answers. Now you read, you will understand. Now we'll move to the another concept. Now we will talk about the uh, now we will talk about the stem cells concept. See, stem cells are those cells which can become a specialized cell. Okay, these are very let's say primitive cells, unspecialized cells. Okay, when you took a birth, you were a single cell. Now from the single cell you have grown up a big person, you have millions of cells in this. Okay, single cell has been developed. Okay, this single cell is nothing but the stem cell, which has a pluripotent property. Pluripotent means it can take, single cell can become anything: eyes, a heart, kidneys, livers, anything. Once it attains one thing, let's say liver, now this thing will become a specialized cell. Okay, it can develop only into the liver, it cannot develop into the liver cell, cannot be developed into the kidney. But during time of your embryo formation, here you can be developed into the anything. These are the unspecialized cells. From this cell you have developed into this thing. Now let's say let's say these stem cells have different kinds of the functions. Now through this studying your embryo, now I can use this embryo for the research purpose, because through this embryo I can culture the cell from here. From here I can use this cell and I can develop a kidney. Okay, I can develop the liver. Okay, for the therapeutic purposes I can use it. Or else I can use your cell. Okay, I can use your cell and I can put it into the mother. Okay, just like a process of the cloning. Okay, I can put into the nucleus of the mother, and I fertilize it. Okay, then let's say you have the heart problem. You your heart is broken. Okay, your heart is broken, you have the heart problem. Now I need to treat your heart. If I take someone else's heart, that person will also be heartbroken and die. Okay, yeah. Let's say what kind of medicines it will work. Now I have taken your cell structure, now I have transferred into the embryo, and it has been fused. It will become a pluripotent stem cell. Now I am experimenting these things. So now what is happening now, I can develop a kind of medicine that is suitable specifically for you. Or else I can develop a heart, new heart, Atmanirbhar. Your own heart is being transferred to yourself. Okay, so this is the thing. But what is happening here? One more life is being created. So there is a ethical problem. So things are clear, simple, easy. So nothing but one thing: stem cells. These are the primitive cells. Okay, unspecialized cells which can take a specialized format. Uh, Use it for the therapeutic purposes and any research purposes and all. Currently, we use stem cells only for the R&D. Let's say you might have seen the lizards where the tail is gone. Okay, the tail is gone, and again it will regrow. Okay, it's not for you. If I remove your hand, it will not regrow. But let's say in the future, if we use the stem cell concept, and you will also become like a Terminator, regrowing your hands. So this is the one concept where the stem cell currently it is using for useful only for the research purposes. There are certain guidelines, ICMR guidelines, that are being released how the cells, stem cells should be done and all. So that's it. These are the various concepts in the biotechnology. No, nothing, nothing technical in these things. I think I've covered all the basic things about the immunity system, biotechnology. No, whatever the things that are there, everything we use the same concept. Let's say now in our body we don't have everywhere these stem cells. Okay, our cells has become the specialized cells. Now there is the person who has made this induced stem cells. Okay, your somatic cell is a kind of specialized cell. Okay, it cannot generate the heart, kidney, liver, anything. Okay, they will have one specialized purpose. Now this thing has been sent back. Okay, it has been developed into the ancient form, pluripotent. It can now, it has made a stem cell, induced stem cell technology. It is also in the news. This is that concept. Okay, so all these concepts are based on this only. So now we have seen cloning, three-parent baby, stem cell concept. Totipotency is also same. Okay, totipotency, pluripotency. Mm, these are the things I think I have covered everything. There was also one more thing in the news that is called a polymerase chain reaction test, or else ELISA test. Okay, so what is happening here is, let's say now we have to check whether you have the corona or not. So it will take 14 days to show the any kind of infection systems. Initially, the virus is very less in number, so it is not easy to study. Okay, and they are using the you might have heard in the news antibodies test. Okay, in the starting itself I have seen, as a response to the immune system, your body's your blood will develop the immune system. So generally when the dengue and all is come, then your immune system will be down. WBC, they will check up the blood count, WBC count, so that they will understand. Okay, to understand how your, based on your immune system antibodies and all, they will understand whether you have been affected by the antibody virus or any kind of infection or not. So that is one thing. Now polymerase chain reaction, what is happening is your cell will be multiplied into n number of the copies. So you need not wait in for long period of time, so that all these things in antibodies and all grow bigger, and then you can able to see. In the starting itself, you can measure this, you can multiply your cell copies, and then you can easily identify them. ELISA is a test again based on the antibodies. Okay, so these are certain things that are in the news. PCR, it is also used in the HIV. Okay, so that's it. Now you, whenever you see any basic topic on the anything on the biotechnology, you will be able to understand this thing. I've covered every basic thing. You know, you just go through. You don't have to get a lot deeper. Just go through DBT, or the Department of Bio, whatever the project that are CSR doing. Just 2 3 lines. I've given you basics. Nothing technical, nothing conceptual under this. I have given you broader overview. Okay, just now you should understand. Now you try to solve even the questions. You see the questions. Okay, they have asked only these kind of basic things. Okay, what the DNA is made up of, what kind of what is the somatic cell transfer, what is cloning. Okay, what is based on this thing, what is a transgenic animal. Okay, can we transfer DNA from plants to animals? Okay, what is does your DNA have the carbon, oxygen, hydrogen atoms? These things, these are the you go through your previous year prelims bits. Okay, these are the certain basic things that has been asked. That's why I'm asking you to focus well on the science and technology environment, and then economy. These three things, and then international bodies. If these things are very clear, your claims will be taken care easily. uh, Please just study well. Okay, if you have any issues, let me know. Thank you. Study well. Bye bye, take care.